I think we should get started. Can you press the hide option at the middle of your screen so that this does not distract from the presentation? Perfect. Sir, you can start. Sir, we can start introducing our resource person.
Good morning to all. Mm, So, it is my pleasure to introduce you Dr. Javed Ahmed Mir, Principal Scientist, Central Institute of Temperate Horticulture, and he is presently the head of the CPP, the flagship program uh, which was started, which is directly monitored by the PM directly, and which is a DEEM project of India, where we have planned to make our plant safe and free from any viruses and best quality produce planting material. And I am happy to share that Dr. Javed is heading that organization now at the because 5 centers have been introduced, sanctioned pan India, and one of the center have been sanctioned at Srinagar. So it is my pleasure that I am introducing my brother, my mentor, and my colleague, Dr. Javed. Basically, he's a biotechnologist by degree, but by profession, by his expertise, he is a one-man army. He is a great fruit scientist, a fruit expert, we can say, a pathologist, entomologist, soil scientist, breeder. So I have been the pleasure uh, of working with him from the past 10 years, and I can understand how much, what is his potential. So it is a pleasure that he is here for training. At the end of the this training, we have planned that we will make the best faculty available from Pan India. So you can understand that Dr. Javed is a well-known personality throughout the uh, IC, not only ICR, but all the institutes, all the universities, and all the organizations working in temperate horticulture. So presently, he is working on very important aspects also, the development of hybrids, which I am also the part, and we have been successful last year by releasing three scape-resistant varieties, which are first from our institute, and scape-resistant varieties of apple. So I am I am satisfied and I am hopeful that you will get maximum benefit from his lecture. But I kindly request all participants, because this or like NADC, DCL Baramala, ICR 388, and also Dr. Shoaib KVK, we have tried to make the best faculty available for you. But it is now up to you to take the best benefits, maximum benefits from these faculties. So I request to you all, and I personally hope that you will listen, you will learn, and at the end of this session, I am hopeful that you will interact with the expert and you will ask your queries, so that your queries will be redressed, and you will get maximum benefit from this wonderful and knowledgeful and lecture from one of the best experts of our country. Thank you, madam. Back to you.
Thank you, Wasim. Dr. Wasim, it was really exaggerated introduction. Actually, I'm not so much expert as much you are and other people are around. But thank you so much for introducing me to the faculty which are participating in this program. May I know how much time I have to present this breeding? Because this is a very important topic. Everybody of us should know how to breed perennial crops, particularly fruit crops. So we can discuss because we are involved with this from last 20 years. So may I know how much time I have? Is it 1 hour or
So
more?
it is 1 hour. 1 hour it is, sir.
1 hour. Okay, okay. Accordingly, should I proceed?
Yes, sir. Yes, sir. You can carry on.
Okay, thank you so much. Let me start with the actually breeding programs in perennial crops. I will discuss here a few crops, particularly apple. See, we started our breeding program long back in 2007, 2008. And at that time, we thought that the breeding in perennials, is very different from what we do actually in annual crops. See, in almond crops, we do pre-breeding, wherein we produce homozygous parent lines, and then we carry out the hybridization, and we get heterozygous F1 hybrid. And from that F1 hybrid, we go for selfing or back crossing to induce the homozygosity for 3, 4, 5 generations. And at the end of 5, 6 generations, we get homozygous recombinant inbred lines, which are homozygous. And then we go for the selection of those homozygous recombinant inbred lines. That means pyramid goes from minimum diversity to maximum diversity. That means F1 is completely uniform there. Then we create segregation lines, and we finally increase the diversity there in perennial crops, but in annual crops. But in perennials, like we have apple, pear, almond, walnut, they are all heterozygous in nature. We have done the sequencing of these crops, and let me tell you, in apple we have 60% heterozygosity. That means if there are 100 genes, 60 genes are heterozygous in nature. That means when you sow these genes by seed, you will get segregation, complete segregation there. So you won't do breeding by that pattern, what you are doing in normal animal crops. So in this case, you have reverse pyramid of breeding strategy. That means when you go for the crossing between two parents, let it be in apple. If you do the crossing between parent 1 and parent 2, you can get segregation to a limited extent, and that segregation itself is the advanced segregation in annual crops. My point is that when you plan a breeding program in perennial crops, you have to select two parents, and the number of crosses you have to select at the maximum. It should be maximum. Let us discuss that point in next slide. I will show you how to plan breeding in perennial crops. That's very important thing in perennial crops. Otherwise, the rest of things are in advantage with us over annual crops. So these were the four crops which we selected at the baseline in 2008. Apple was selected, pear was selected, almond and walnut was selected. When you select a crop for any breeding purpose, you should know the trait which you want to improve. In apple, first of all, we selected our own variety, that's Amri. Why? Because a lot of work is being done in apple. You see, these days, thousands of apple varieties are flooding from other countries to our country. So you cannot compete with the rest of the countries. But if you have some advantage, we do have some advantage. We do have our own Amri varieties, which is an advantage over other varieties available in rest of the world, because our Amri is having better shelf life and it is rich in aroma. So what is lacking in that Amri, we want to improve upon that. It lacks a beautiful color. It has a moderate scab resistance. It's not exactly a regular bearer, and also its maturity is little bit late. So we want to improve upon these traits so that we can get an ideotype, the best genotype with these four traits and its existing two traits that we have achieved. I will show you in advanced slides also. That was our first objective for apple in Amri improvement for these four characters. And we also do the improvement of other commercial apple cultivars, because we do have a lot of apple cultivars available here. Like we have Delicious Group, Gala Group, and Fiji Group. Although they are very advanced groups, they possess some desirable traits, but still they lack some traits which we need to improve upon, like scab resistance is required. And also, for example, in Gala, we need to advance its shelf life. In Fiji group, we need to induce the color in there, because proper color is not coming in the Fiji group. And third, very important objective in our apple breeding program is the development of our own pollinizer. Because, an apple which has self-incompatibility, gametophytic self-incompatibility, when we lay down an orchard, we need to add 20% of pollinizer into it. When we add 20% of pollinizer, it means a lot of plants need to be planted. For example, in per hectare, we are planting 3,333 plants. 20% of that means 660 plants almost. So if those plants belong to a variety which will not give you a good market price, it is a loss to it is a loss to our orchardist. So our pollinators should possess the fruits which have very good consumer acceptability and market demand. So Golden Delicious, although it's a very good variety, but it has less consumer acceptability over other varieties. So we need to have a different ideotype. Maybe with Golden Delicious background and more colored element, and it should be result free. This is a problem with Golden Delicious. That is our breeding for pollinizer development in apple. In pear, pear, we have two groups, Asian pear and European pear. Asian pear, which is a maybe we can say non-climatic and moderately climatic in nature. European pear is a climatic in nature. That means European pear has a less shelf life, but Asian pear has a more shelf life. But Asian pear does not possess a good appealing color. So also, European pear is completely incompatible. It has 100% self-incompatibility. But we do have self-fruitfulness nature in Asian pear germplasm. So in this case, we are going for interspecific crosses between Asian and European pear to develop an ideotype having higher shelf life, better color, self-fruitfulness, and also additional traits like disease resistance. And we have developed a huge population, and we are with some selections of our these segregating populations which possess all these traits, and now we're in a way to release them. And in almond, almond faces a big problem of winter, this spring frost, because it gets very early flowering. Among these crops, almond is the crop which flowers early, and if at that time there is a low temperature, then there is the problem of frost damage. So we need to have late flowering almond cultivars. And also in almond, self incompatibility is again there due to gametophytic self incompatibility, which is induced by S alleles. And if we have some almond cultivars with very good commercial traits and self fruitfulness nature, that will be a eureka for us. And for that matter, we have imported germplasm which are self-fruitful, like Tuno, Independence, Sasta. These are the varieties which are self-fruitful, and we have generated the populations, and we are selecting our cultivars with these late flowering traits, self-fruitful traits, and traits which are contributed towards nut and kernel quality. And in walnut, our state, we can say the J&K state has the world's best walnut. Even our institute, we do have released one variety, CITH walnut one, which bears a fruit of 28 grams. You can check the literature, you can check the internet if there is any walnut size which is greater than 20 grams. No walnut is greater than 20 grams. This point of time, one walnut variety that is known as Chandler is popularizing like anything, but its size is only 14 grams. That means our country, we possess a walnut with the best quality with respect to nut size, kernel yield, kernel qualities. But the problem with our walnut is that our walnut is terminal bearer. There is no lateral bearing walnut available so far in our country. But recently we have released one of them. But still, the most improved, most good quality walnut cultivars, we want to introduce introgress lateral bearing trait into them. So our breeding program is focusing on introduction of lateral bearing trait into walnut. See, this is a very important slide. One who is interested in the breeding of perennial crops had to think like this. If you have started, I'm showing the story of our one variety which we have released recently, CITH Prime. It's a hybrid between Prima, which contributes to scab resistance, and Ambri, as I was telling, our own Ambri, we want to improve upon for scab resistance. See, we started program long back in 2009, 2010, and we germinated the seed. when you have to start a breeding program between A and B, don't cross 10 flowers of A and B. Don't cross 100 flowers of A and B. Don't cross even 1,000 flowers of A and B. Carry out the pollination crossing of at least 20, 30,000 flowers of A and B. So first of all, when you think of breeding, you should have got a planting material sufficient in flowering, which can give you pollen, sufficient pollen load and flower count, which will augment your breeding program at the start. You cannot start a breeding program in perennial crops with 2 plants of each. You can introduce 2 plants and think of starting breeding program. No, it's not like that. You have to import planting material, for example, of X variety. If you are importing only 2 plants, you multiply those plants first of all and get a population, sufficient population, wherein you can get a pollen load as and also flower count equal to maybe 20, 30,000 flowers. And think of that. Think big, then you can achieve a big. Otherwise, it will lead to limited achievements. So let us start a crossing between A and B. Let us start, keep in mind, at least 10,000 flowers, which we have started. We have started with 20,000 flowers between CITH, Ambri 1 and Prima. And we could germinate 10,000 seeds. 10,000 seeds were put for germination in 2014. And from 10,000 seeds, we obtained 7,000 seedlings in 2016. And in 2018, from 7,000 seedlings, we could get 4,000 graftable seedlings from them. And from 4,000, phenotyping was done in 2020 for 1,100. Because there's a mortality and some did not bear the fruits and many other things happened in between. So at the end of the day, in 2023, we had only 400 to 500 plants available with complete fruit quality, trait data, genotypic data available. I can say that 500 plants for me, for our team, including Dr. Wasim, including Shoaib Kirmani. Dr. Shoaib Kirmani was the farm in charge during these activities, and he handled these breeding material. He was handling this 4,000 rootstocks at that time when he was farm in charge. And handling of this breeding population is a big ask because you have to maintain this. And at the end of the day, in 2023, we have 500 plants. I can say these are sufficient for breeding purpose. If I would have started with these 500 plants at the time of germination, then we would have got only 10 plants right now, which was not sufficient which would have not been sufficient for our breeding program or phenotyping. So think of like this. So we can say 2010, 12, we have 2014, we did this germination studies. That means we have done crossing in 2012. From 2012 to 2023, we could get the variety. That means at least 10 years of journey is there for a varietal development program in any perennial crops. And if it is rigorous and complete, every step goes well, then it is 10 years cycle for this breeding program. This is a glimpse of this breeding populations, germination of hybrid seeds. We started 10,000. This is the raising of seedlings. You can see the number of seedlings going on. This is the phenotyping of these seedlings, grafted plants of crosses. And on right side, you can say the population in bearing, which you go for the phenotyping studies. Let me tell you how we go with the CITH Ambri 1. As I said at the very beginning, that in our own Ambri, the Ambri is a clone. At right this time, if we say Ambri is a mixture of population. It's a huge population. One Ambri will be of different type, other Ambri plant will be of different type. So first step in your breeding program of anything, if you will start a breeding of delicious group also, you have to select your delicious, because delicious has also become a population. Golden delicious or red delicious, it is again a population, because so many farmers have raised the plants from seedling. So segregation occurs during the course of its commercialization. And when segregation occurs, your true to type nature goes. It does not remain there. It is only possible through vegetative means. So right now I can say red delicious, golden delicious, Ambri, they are all populations. You don't have any pure or homogeneous line across your Kashmir. Right now, if you are actually importing planting material for new varieties, they are homozygous because vegetatively propagated right now. Because if any farmer will propagate them by seed, it will get again a mixture. So my point here is that Ambri, whatever available in our Kashmir Valley or in our India, it's a mixture. So first step was to select the best out of them. And we selected best out of them. That was CITH Ambri 1, which was registered with NBPGR, and it was evaluated completely. And this particular Ambri clone is being used for hybridization. We did hybridization of this CITH Ambri 1 with Prima on one side for scab resistance, with the Gala strains, Gala Shinigo for color development, with Redland columnar varieties for columnar nature, and with Oregon Super for regular bearing. And we did this crossing of about 10,000 pollinations. And then after that crossing, you can develop a population for evaluation, maybe for quality evaluation or resistance evaluation. And after that evaluation, you can identify a superior genotype. And that after identification of a superior genotype or hybrid, you can say you have to go for the multi-location trial if you want to release that variety at national level. Go for the multi-location trial. For our perennial crops, multi-location trials include our J&K. You can go for Himachal, you can go for Uttarakhand, or even you can go for northeastern states like Arunachal Pradesh. If you will, there are two things. Those people who are involved in breeding may kindly listen these points carefully. If you will, any hybrid variety, anything, if you will evaluate your hybrid or variety in your own state, that means J&K, you can release that variety by State Variety Release Committee, and it will be notified by Central Variety Release Committee, but only for J&K state. It is not like that. You have to go for the multi-location trial strictly for release of variety. No, you can release the variety if you evaluate it at a single location also, but it will be restricted to your own state. But if you have multi-location trials, if you evaluate your variety at least 3 locations for J&K, Himachal, Uttarakhand, and you have the data for 3 years for that variety evaluation from these 3 stations, then you can release the variety through SVRC and CVRC. Your notification will be from CVRC, Central Variety Release Committee, for J&K, Himachal, and Uttarakhand, and all over India. If it is more than 3 states, all over India. So that will be released at national level. That is the release pattern. This is how it goes. You cross Ambri and Prima, you develop population, F1 population, and evaluate over years. This F1 2000 is those which are in bearing. And these are the fruits. We see the segregation pattern. You have a lot of segregation pattern. The advantage in perennials is, listen my point, advantage in perennials is, if it would have been an annual crop, after F1 we should have gone for selfing or back crossing, again selfing and back crossing to retrieve the recurrent parent back cross, and selfing to induce homozygosity thereof. 5-6 years selfing was required in annuals, but in perennials no need of further crossing. You have to develop only F1, but the number of initial crosses should be high. When you do the initial cross number high, you will get F1 population. Just use that F1 population for grafting, and you will get a bearing population and evaluate that bearing population. This is the way how to evaluate the bearing population for fruit crops, and from that evaluation you can get your genotypes, the best hybrids, and those are the hybrids which you can go for the release or registration. And also you can use that F1 population, the population which is in bearing, you can use that population for genotyping, fruit quality analysis, and linkage mapping and QTL mapping. Suppose you have done cross of A and B for trait X, like we have aroma trait in Ambri, and you have evaluated your whole population for aroma trait, and also you have done genotyping of your whole population. Now where does that aroma trait lie? It lies at which chromosome, at which place you can identify, and which are the markers which are linked to that particular trait. You can identify QTL. Then for fine mapping, you can go for fine mapping of that QTL, and you can link the exact marker which is responsible for that aroma. So identification of markers is very important in these biparental populations through QTL mapping. And if anybody is involved in breeding program, a biparental population development program, and he or she should go for linkage mapping and QTL mapping. And after identifying a desirable QTL using some internal mapping pattern, he or she can go for the fine mapping of that QTL, and finally identifying the marker which is associated that particular trait. And that marker can be used for marker trait association studies in future work or any breeding or improvement program. One more crossing of our CITH Amri was done with red line. We introduced these columnar varieties from Netherlands. These are the mini plants. See, one plant bears 200 to 300 fruits. We could get 220 fruits from one plant after 3 years of this grafting. And also, we can get fruit in the next year during the grafting. इसी साल आपने grafting की, इसी साल आपको fruit मिलेगा. So these kind of plants have advantage. They are scab resistant. They are low chill. They require only 100 hours of chilling. Chilling means 2 to 7 degrees centigrade accumulation, which is available in Delhi, which is available in Delhi, Haryana, Punjab, anywhere. So these plants can be grown in that places in terrace farming as well. छत पे लगाओ जमीन की जरूरत नहीं है, because you can grow them in pots also. Urban horticulture, because people in urban areas don't have land, they can grow such kind of plants in pots. They can give them fruits as well, and also aesthetic value as well. And one more important thing for a breeder who want to breed, who want to use the speed breeding. So these are the candidates, because they will give you the fruit next year. So these kind of plants, these kind of genotypes can be used an intermediate parent, which can induce the precocity in your population, and you can evaluate your population in next second year only after your crossing. So these at least I said that in 10 years you can complete your breeding program. You can release variety by using these as a intermediate speed breeding partners. You can develop a variety in 7 to 8 years only. So that is the beauty of these columnar varieties. We do have also breeding program, introgression of traits from these columnar varieties into our Amri as well. One more thing is breeding through mutation. Mutants are wonders. Mutation is a wonderful thing which you can do for traits like fruit color, for those traits which are usually monoallelic, which are not polygenic. Polygenic traits usually are not much affected by mutation, but oligogenic traits, which are contributed by few genes, they are getting influenced by mutation. Color is one of them. So our program was to induce the mutation in Amri, and we develop a mutant population of more than 1,000, 2,000 these sticks we used, and more than 6,000 buds they had, and they were exposed to gamma radiation of 30 and 40 grays. And at last year we had about 300 these mutants which were in bearing, and we evaluated these 300 mutants for color traits using RHS color chart, using Hunter Lab LAB color chart, and also for carotenoid metabolic pathway studies. And we identified 2 mutants which were having all the properties retained from our Ambri cultivar, and they possess a very good high intensity color values as well. So this is one of the beauty you can use in your perennial breeding programs, because gamma radiation plays a very important role. You can go for chemical mutation also, but chemical mutagens like AMS or UVMS, they are although very good, but little bit regeneration problem is there. When you expose your buds to those chemicals, you may get little problem in your bud sprouting, but you have to standardize the dose. It should be less than 5% or 0.2%. You can play with that also. And mutant population study thereafter also. You can go for tilling, you can go for whole genome sequencing, you can identify SNPs linked to the change in traits, like we have developed these color traits in mutant lines. We are in a way towards tilling or SNP genotyping of these mutants to identify actual transition, whether this is transition, transversion, or deletion, or insertion. Insertion won't be there, whether it is deletion or any other thing which caused this mutation. So this is very important one area which you can apply for induction of this traits into your perennial fruits. But only thing is when and how to do. Two things are there. You can do you can induce mutation in buds during the budding season. Usually we do budding in August. So you can use the mutation at the time of August, and you do mutation in gamma chambers, bring them back in some cold these packs, and directly go for the budding. Or you can use in vitro mutagenesis also. If you have standardized the tissue culture protocol, you can use your explants or shoot apical buds or your vegetative proliferants. You can use auxiliary buds also. You can take directly those bottles to gamma chamber, expose them to gamma radiation, bring them back, go for subculturing, go for the evaluation of your mutation as well. It is better to go for the mutation using this in vivo technique, which we did with the budding, because you can get plants in next year or only, but in tissue culture it takes lot of time. Otherwise you have to go for the in vitro grafting also. See, once you develop any mutant, once you develop any hybrid, anything, this is just my one screenshot I want to show you. Evaluate your hybrid variety, any mutant thoroughly. Evaluate your mutant, your developed variety in comparison to parents, one thing, the parents you have used, and also in comparison with the most commercial cultivar available in world at that particular time. When you evaluate your variety, mutant, or anything you have developed with the parents and commercial cultivars, if there is a advantage of any bioactive compound to it, it will add the value to your this hybrid, to your variety. Similarly, we have done evaluation of all the bioactive compounds available in apple, whether it is quercetin, rutin, catechin, epicatrin, ferulic acid, chloride. There were so many things. I have shown one screenshot using HPLC. We could identify few bioactive compounds which were exclusively overexpressed in our hybrids. So that will be one of the advantage when you release, register, or commercialize your hybrid, because that will act as a specific marker, specific trait to your variety. So you should not stop by evaluating your variety for yield and quality trait. You should not stop there. You should go in advance. You should evaluate it at biochemical level and also at molecular level. We have evaluated our hybrids at molecular level as well. We could identify the genes which were responsible, the traits we are claiming here. For example, we are claiming traits like sweetness, we are claiming traits like higher shelf life, we are claiming traits like scab resistance. All the markers were identified, all the genes were identified through comparative transcriptome in those hybrids, so that at the end of the day we can say, yes, our hybrids contain these type of principle which is specific to our hybrids only. So based on these evaluation, complete evaluation of these populations hybrids, last time, as Dr. Wasim said, because he's one of the main contributor towards the development of these hybrids and hybridization program, we released variety Sith Umul, which is an improvement of Ambri for early maturity and color. Ambri possesses aroma and shelf life. Now we have hybrid which possesses aroma, shelf life, early maturity and color. One more is Prime. It's a Prima and Ambri. It contains aroma, shelf life, scab resistance, early maturity, and color from Prima. So now we have Sith Prime, which possesses aroma, shelf life, scab resistance, early maturity, and color. Sith Pride, beautiful hybrid, which possesses scab resistance, early maturity, and color. So these trait combinations are now augmented, introgressed into the varieties like Ambri. Now we have improved Ambri with us, with all the traits which our imported varieties possess. So our varieties are even better than the imported varieties because our variety possesses higher aroma and shelf life. And also in walnut, we have early majority walnut, sit early with nut yield, quality, higher kernel, and nut recovery. Sit prolific, that is what I was telling you. We could identify the lateral bearing walnut variety having a higher yield and quality. I will show you. We have released this variety. This is the first lateral bearing walnut variety released from any institute in India. And also Sith Red Gem in onion, we developed a variety and released it with respect to bulb color. It has a very good bulb size and color. And Sith Jumbo is a garlic variety which we released along with our perennial fruit crops, and it has a potential higher yield and a good color, and it's non-bolting. It does not show bolting at all. So this is I was telling about the Amul improvement of Ambri. See, you can see the Ambri in middle and Amul at the below. It has a much better quality than that. And we have also done the fingerprinting of this, and also we have developed a marker for its identification using allele tapping. And also we have done complete metabolic profiling, VOC profiling, and gene identification of this variety with respect to traits we are claiming for. For example, we are claiming for early maturity, we do have marker for that. We are claiming for scab resistance, we do have marker for that. We are claiming for higher shelf life, we do have marker for that. And we do have the quantum of changes available throughout the development of this fruit for that, because we have studied this for all these leased varieties, and we have sufficient planting material available with us right now. We have more than 1,000 plants available, and it has been also distributed among the stakeholders like government development departments. Similarly, in prime, similar this marker pattern, planting material availability. Also in pride planting, and see, this is the pride of one beautiful can see the picture of this pride is a beautiful color, is even better than color available in the imported varieties like Jeromine and this Red Rocks. This is the pride variety, and markers for use for its identification and fingerprinting. And also see the kind of scab resistance we are screening for. We are grafting both parents commercial varieties and our variety on same plant. You can see this Red Delicious completely devastated by scab resistance, and our pride, it has no symptom of scab on leaf or even on fruit. Sufficient planting material available with us for this. This is one of the hybrid which I was telling at the very early slide, the development of pollinizer. We do have Golden Delicious pollinizer, but pollinizers are not having such a good consumer acceptability because it has the problem of resetting. It's very late maturing also, and also some other problems with its acceptance. But we need to develop a pollinizer which possess the traits like higher shelf life, although Golden Delicious has higher shelf life, but it should have additional traits like tolerance to resetting, and its color should be a little more acceptable than what is Golden Delicious. So we developed a pollinizer Golden Snow. It has a very appealing color. Its color is so appealing. When it will be available in the market, people will forget the red color and they will take this creamy color apple, and with higher TSS value and with zero resetting. And this is the pollinizer we have developed, and hopefully we will release this reset-free early maturing or large size fruit pollinizer variety in this year. We have also developed the molecular markers for its identification also. So that was the kind of varieties which we have released. Now you people will say that a lot of varieties have been developed in scab resistance by other people around the world, but the problem with those varieties is that one thing is that their resistant phenomena can break down at any time, and second thing is that their appeal, their size, their acceptance with respect to TSS and other factors is very limited than the varieties which we have developed. Also in walnut, we have developed the varieties. This is the early maturing walnut, and we have sufficient planting material. We have done fingerprinting of that. This is the lateral bearing walnut, first walnut variety which is lateral bearing in nature, and we have it as sufficient planting material. It's correct precision and also been done through fingerprinting. And in almond, we have developed the population using self-fruitful genotypes like Tuno, Independence, and Sasta. We are in a way for development of QTLs and linkage maps. And in walnut, in almond, we have the population developed, but it is not yet in fruiting. But we have done comparative transcriptome analysis to identify the markers which are responsible for those traits. And using those markers, we have identified some genotypes in them which have this corrected traits available, and now those will be validated at the time of their fruiting. And again, walnut. See, walnut, we have different lateral bearing genotypes. One genotype available, which was available with us long back, available with India, that was Sir, but it has only 50% lateral bearing potential. Piney, which has only it is less than 50. Here it is. I have shown greater than 50, but it's less than 50% in our conditions. It does not show about 20% of lateral bearing in our conditions. In USA conditions, they are showing this much of lateral bearing potential, but in our conditions, they are not showing. Chandler, which is 80% lateral bearing potential. We have developed population with all of them. And those populations are ready, and they will be evaluated for lateral bearing potential. If any variety, if any hybrid, we will get, which is having nut size of our walnut series, maybe 28 gram, and lateral bearing potential of more than 50%. That will be a real good achievement for us. And I think I told you that that will be a eureka for us, our breeding program. That's true. That will be a eureka for us to get a variety with nut size of more than 28 gram, more than 25 gram, with the more than 50% lateral bearing potential. Nothing better than that. I may tell you, I was in Uzbekistan last year, and we had a vast exploration visit over Uzbekistan there. We could identify one walnut variety there which gives fruiting in nursery itself. When you graft a plant, whole graftage, 1,000 plants were in graftage, and all graftage pattern was in fruiting. I got surprised, and we brought that genotype through germplasm exchange, because we had one program sponsored by DST with them. And this is the lower, this ideal is the genotype which we obtained from Uzbekistan, and in our conditions it gave fruits in very first year. You can see hundreds of fruits per plant there. So we are using this as well for crossing, and it is having more than 90% lateral bearing potential. That is one of the best lateral bearing potential I have seen in any variety of walnut. So this will be also one of the achievement of ours, and this itself can be commercialized. We are into it because its nut size is more than 14 gram, at least more than Chandler. It's 18 grams, and its nut qualities are even better. We are thinking of this popularization as well. In pear, I told you, pear, we do have two groups. One is Asian pear and one is European pear. Asian pear itself is superior with respect to self-fruitfulness, with respect to shelf life. But the problem with Asian pear is color. Color is the possessive quality of European pears. That is why we are doing interspecific hybridization as well. In pear, Dr. Wasim is the lead breeder. We have started breeding long back. We have developed a large number of populations, and we are interested in development of an idiotype, which is low chilling, because in pear, we have the advantage of area. In apple, walnut, and almond, you have only J&K, Himachal, Uttarakhand, and northeastern states, particularly Arunachal Pradesh. But in pear, you're not restricted to these 4 states only. You can go beyond that. You can go to Punjab, Haryana, Delhi, even surrounding parts in south, some parts like in Bangalore. You can go there, wherein you can get a chilling of 200 hours, because they are low chill varieties. They possess low chill traits, particularly our Asian pear, that trait in Asian pear, low chill, self compatibility, and higher shelf. That's one of the beauty of Asian pears. And based on our breeding program, we identified these 4 varieties. One is Sid Amber, Sid Gold Pear, and Sid Bush. They possess low chill, self-compatibility, and higher shelf traits, and they are under evaluation. Most probably this year we will get through them, and we'll be able to release these varieties. And their release will be done all over India. We'll not restrict ourselves to J&K, because we are interesting, because they are low chilling. We are interesting to promote our pear germplasm to rest of the India, particularly Punjab and surroundings. We have developed markers for them. We have developed a tapping for its identification, with the first report for apple as well, for pear as well. And we have evaluated them with respect to shelf life. And we have seen that there's a significant quality retention in these pear hybrids. we have developed that interspecific crop, which was fresh. After 3-4 months, we consumed it when it was kept at room temperature. Dr. Wasim brought it to me, and he said that taste this pear in the winters. It was winters. I told him that it will be deteriorated, and just show me. Then we cut that pear. It was as fresh as we pick it up, the pear at that particular time from the plant. So many of the visitors visited our institute, and they really appreciated the kind of these hybrids we are developing in pear. And I must congratulate here Dr. Wasim Hassan Raja, who has contributed tangibly, significantly in this pear breeding program. And right now we have about 6 pear hybrids which are ready for commercialization, release, and promotion and registration. And also we have started root stock breeding program in apple. Root stocks, almost 100% of root stocks, clonal root stocks in particular, are being imported from rest of the countries. And nowadays, we are up to high density plantation. And if per hectare, we are growing 3,333 plants. So you need to have 3,333 root stocks for them. For each one variety, you need to have each one rootstocks. We are importing in millions per year rootstocks, and we are paying a lot of money to other countries, and they are doing nothing, only multiplying the rootstocks, the kind of rootstocks which are already available with us. Now, what we are doing, since these M9 clonal rootstocks, they do possess the quality like clonal propagation, precocity, induction, and induction of developing. But they lack some qualities, like they are not tolerant to drought. They are not tolerant to some disease. They do not have any tolerance to root rot. And very few rootstocks have been identified which have tolerance to rootstock. But now we know that Malus baccata is very tolerant to rootstock, and it possesses traits like the tolerance to our drought as well. So we start breeding programs. We cross our Malus baccata with these rootstocks. We develop a lot of populations. And Dr. Wasim Hassan Raja, again, leading this program of clonal rootstock development, a rootstock breeding program. And we identified the rootstock populations, rootstock hybrids, which were having the potential of multiplication. See, if our Malus baccata hybrid, if we are hybriding our Malus baccata with M9 or any other rootstock, if that population, if that hybrid possesses the clonal multiplication potential, that means it contains the background of Malus baccata 50%, and it has the multiplication potential. Then what else we need? That is the best thing we got. We got very strong multiplication potential of Malus baccata hybrid with Bud9, and that hybrid possesses the traits of Malus baccata like drought tolerance and other things, and clonal multiplication as well. So we are evaluating these apple root stocks for wood resistance. Again, this is the initial screening we are doing. We are evaluating these hybrids against white root rot, and we got some initial glimpse that some 6 root stocks we observe that they grass. So we'll be evaluating them, and in future we are thinking that we will get a 6 series of rootstocks available which possess the traits of clonal rootstocks like clonal multiplication, precocity induction, and developing induction, and the traits from baccata like drought tolerance, disease resistance, and multiplication potential. That is what we aim for. That's our ideotype for rootstock. So I must thank you for this listening my presentation, and actually my aim was to enlighten you about the program, what we are doing, and if somebody is interested in taking up this perennial rootstock breeding program, so what kind of strategy they should adapt for. So this is a reverse pyramid. Actually, normal pyramid breeding program occurs in annual crops, and this is the reverse pyramid breeding program which occurs in perennial crops. So thank you so much. Thanks for listening my presentation,
Thank
and
you,
thank
sir.
you
Thank you
for
for presenting
organizers
beautiful.
for giving me this opportunity to interact with you, to show our achievements to the people who are involved in the breeding program. I must thank the organizers as well. Thank you so much.
Anybody from the participants who want to interact with Dr. Saab? So there was a one question from one participant about walnut variety.
Yeah.
She wanted to know Pusa Kohar. Is it lateral bearing variety of walnut or not?
No, we do have. This is the first lateral bearing walnut which we have identified that is prolific. And we have available right now imported varieties which are lateral bearing. Only one is, I told you, Sir and Piney, they were very early stage. Now Chandler is coming, which is having more than 80% lateral bearing potential. And also, lateral bearing is nothing. It is a bearing when you get a female flower from auxiliary bud. Sometimes you have lot number of laterals. If you have lot number of laterals in your walnuts, but they are not from auxiliary bud, that is not lateral bearing. If you have direct stigma formation from your auxiliary bud, that is the lateral bearing. That potential only lies with Chandler and other very few genotypes.
Okay, sir. Amir, you want to ask something?
Good afternoon, sir.
Good afternoon. Very good afternoon.
Thank you, sir. It was very, thank you, sir. Thank you, sir. It was very interesting session from you, sir. But one thing, sir, I want to ask you, and I want to add this session that in Kashmir we have, especially, I'm working with high density apple orchards. Especially, basically, I'm covered from first Kashmir, and I'm working in field. Sir, we have a common problem here from last 2 3 years. So that is white grub infestation. That is very fastly damaging our plants, especially newly established high density orchards. So, sir, we should have, yeah, or we must have some strategy for this to breed or to have to develop some root stocks like we have different kinds of root stocks like having woolly apple resistance. Sir, can we have like that something strategy?
Uh, Actually, people really ask these. This is a very interesting question. Development of resistance against insects. If you have insect which is having sucking type of mouth parts, which usually affect your tender leaves, there we can think of resistance development by enhancing the lignification and enhancing the suberization of leaves. The phenomena kind of mechanical resistance we can develop against the insects. Or see, insect resistance was only possible through transgenics because in our plant system it was not available. That is why BT cotton was taken into account, because BT gene can directly kill the insect through other active procedure. Otherwise, we have only passive mechanisms for insect resistance development. That I told you, we can decrease the succulency in leaves by enhancing the suberization and lignization. Otherwise, insect resistance is a tricky. It is very difficult, very extremely difficult to develop. But a tolerance can be enhanced. You can induce the induction of some exudates from your roots, which can deter the insect attack to your roots. Otherwise, insect resistance development is very difficult because there is no candidate gene or there is no gene analog available against the insect resistance in the plant system. That is only the problem. One thing, one problem with us. Again, Amir, we can screen our germplasm, whole germplasm, because in apple, for example, we do have 300 varieties. We can screen these varieties under controlled conditions against white grub. If some variety has some tolerance, possess some tolerance, that variety can be used as a donor for breeding program. Otherwise, so far, there is no variety identified which is tolerant against the white grub. Screening is the first step which we should do first.
But nowadays we are having severe insect infestation. But in our earlier days we were having here scab, alternaria. But these days we are having very severe insect problem. If I am this time currently this time I am in an orchard. This is having MM111 and it is very much infested with green aphid as well as leaf miner. So we are having everywhere this year and last 2 years we are having aphid infestation very much.
That's true.
Severe repeated are not taking.
That is true. Green apple aphid is your young shoots, which are
Yes, sir.
newly growing shoots, because particularly our this high density plantation, which has more young shoots distribution across the your orchard, across the canopy of your plant, you will get green apple aphid continuously. And also, this leaf miner, it was maybe quarantine issue or any other issue, it was we're not aware of right now, but it is a new pest to our society. And also some changes in temperature patterns across the phenological development of the plants happen when they, if some phenological stage of plant is susceptible to your insect, and that particular phenological stage is giving the temperature which is conducive to your insect development, that becomes a havoc. And that happens this time with this green apple aphid, because temperature rises in April, and during April your leaves are more tender, and at the time of leaf tenderness your temperature is also conducive to insect development. So phenology matches phenology and your damage is higher. That happens due to change in climate. That's the problem.
Thank you, Ahmed. Anybody else? I think there are no more queries from any participant.
So, Dr. Shoaib, if
Yes,
somebody,
sir.
because most of there were 35 participants, maybe 30 participants, maybe
35 to
few
40%.
who were associated with the breeding program, they can contact us anytime. We can have collaborative programs, no doubt about that. You can take their contact number. If anybody is interested in breeding program, we can work together. We can get suggestions from them, and they can take some sort of
Surely.
experience which we have already with us. Then we can work together because all of them are faculty itself. So I must appreciate them that if somebody is
Yeah.
working on parent breeding program, because it is very tough and very useful also on sometimes.
Thank you, sir. Thank you, sir. Thank you
Thank
for
you so
giving
much.
your precious time, sir. Despite of being a holiday, you spent time for this. Thank you very much, sir.
Thank you, dear. Thank you so much. Thank you, dear.
Thanks to all participants.
Okay.
Thank you.
Is this session completed?